questions in the chat, I am happy to answer the questions. But I want to start because there was a question that was asked in yesterday's session, which I didn't manage to get to. Hi, Iris, how are you? Just going to do a quick thing on the secretion of insulin because I think that's an important one. But how are you? Let me just do this. If you want to join me, you can do. Right, so let's have a look at the secretion. Do you have any tips on how to answer evaluation questions? You really struggle with those. The minute you see an evaluation question, the first thing you need to do is remember that it's pros and cons, things that support, things that are against the process. Sarah, yes, give me a second. I will cover that as well. But that's going back to the evaluation. That's the one thing I would say. It's funny, actually, getting excited about the exams is a good thing because then you are ready to move on. But let me just quickly throw in a bit for Sarah because you've asked it, and that one I can do quite quickly. I feel like there won't be that much on the kidneys. Not that they won't ask the questions on the kidneys, but I don't feel like it's going to be a huge part because they've done it for the last couple of years, a lot on it. So if they are asking any questions, it will be specifically looking for your understanding linked to other topic areas. So I'm not sure it will come up, but let me just put a quick one in there for Sarah. So you want to know, explain the water reabsorption loop of Henle. So hopefully, when you are looking at the loop of Henle, you know, if I just bring this in, if I'm looking at that loop of Henle, let me go back up a second. I've got my different permeabilities to water. So basically what we've got to remember is we have cells, and those cells have the phospholipid bilayer, the membrane, which is hydrophilic heads and hydrophobic tails. So therefore we've got the hydrophilic parts and the hydrophobic region. So the heads point on the outside, they orientate themselves towards the water, and the tails orientate themselves inside. So the only way we can get things to be able to move across is by having the right type of channels in there. So the fact that we've got different areas with different permeabilities means that you have your descending limb permeable to water, so they've got aquaporins, and your ascending limb impermeable to water, so they do not have the aquaporins there. They don't have the channels to allow the water to move across. So we know that water can move across the partially permeable membrane by osmosis, that net movement. So you've got to keep that in your head. So what's really important is we have this countercurrent multiplier. And what that means is basically, if you start, we always start at the ascending limb first rather than the descending limb. And the reason we do that is because it's all about you understanding the fact that the sodium ions are going to be moved, and those sodium ions are going to be pumped out of the filtrate fluid into the medulla. So the medulla interstitial fluid is basically the tissue fluid surrounding each of these loop of Henles. So basically we're talking about the fluid which is within the medulla region of the kidney. So when we actively pump the sodium out of the filtrate fluid, we are therefore lowering that concentration inside the medulla. So you can quite clearly see we've gone from a 200 to a 400. So we have got more solute, so a high salt concentration in the medulla. This is why it's active transport against the concentration gradient. What we have to understand as well is that by doing that, by having this high salt concentration in the medulla, what you're then going to have is the reabsorption of water. Water will move by osmosis into the medulla interstitial fluid. So this is important to remember. We're moving the water out of the filtrate and into the surrounding fluid of the medulla. So the descending limb is the only one that's permeable, the ascending limb is not, and that water movement is out by osmosis down that water potential gradient. So that's important for us to note. But then what we have to remember, if I bring this back down, then what we have to remember is when we're looking at the loop of Henle, we know the loop of Henle is in the medulla. We're talking about the medulla interstitial fluid. So basically, if I do this, all this stuff here is the medulla interstitial fluid. Okay, so therefore, if I understand that's the medulla interstitial fluid, what I should then understand is when we talk about reabsorption of water, we talk about it to do with the collecting duct. So ADH, antidiuretic hormone, is going to be released from the pituitary gland. It then travels down to the collecting duct, and then it causes the collecting duct to increase the reabsorption. Faiza, what I'm doing today is I've got an hour I could jump on here. So I'm answering questions from anyone who's in the session and going through content or exam questions. I'm kind of picking a bit of both. I did stuff yesterday and I don't feel like I got through enough. So I thought since I have an hour, I'll jump on here and do a session. So I'm kind of mixing and matching whatever people are looking for, really. But I did want to cover this topic area when it comes to the release of glucose, because I have a feeling, I don't know, it's really hard because I've got different thoughts in my head about where they're going to go with the questions. People always ask me about the predictions that I have, and they change every day because they could go any way they want with these questions. So I just don't know. But if they're going to go down the glucose and hormonal releasing, they could link it to the fact of the insulin secretion, because that is one big topic area. Sahib, I will answer that after I've done this one because I just want to cover this. So yesterday we talked about the fact we've got our sodium potassium pump, and we've talked about the resting potential. But one of the things that we need to remember, it's all about having high potassium and low sodium. So that's really important. So if we now look at a cell, and this time we're in the pancreas, and we're specifically looking at are cells which are involved in our insulin secretion. So because we're looking at insulin secretion, we're looking at our beta cells. So if we have low glucose concentration, you should know the sodium potassium pump is always going to be pumping sodium and potassium in the specific directions. And therefore, we're going to get a high potassium concentration inside. Well, that makes sense. That's regardless of where we're talking about, whether it's the proximal convoluted tubule, whether it's the beta cells here, whether it is the neurons, we're talking about cells and cells having that resting state. So when they have a sodium potassium pump pumping the sodium potassium in different directions, and we have a high potassium concentration, we know that this is a cell which has a phospholipid bilayer. We know therefore potassium has a positive charge, that the potassium can only leave if there is a channel for it to leave. And so therefore here we have a potassium ATP channel, and this is really important. So this is a potassium ATP channel which is open. This means it's a potassium which is activated by having an ATP binding site. So ATP can bind and ATP can be hydrolyzed in this potassium channel. So it's a potassium ATP channel, and it's open. So if it's open and we have a high concentration, what's going to happen is the potassium ions can leave. Now, what that actually does is that hyperpolarizes the membrane. That takes us away from our resting state down to a position which is less than the resting state. In this case, if we were looking at our neurons and we knew they were minus 70 millivolts, the minute we move away from that resting potential, that resting state, we have hyperpolarized the membrane. Well, what you have to understand is that you've got voltage gated calcium channels, and these voltage gated calcium channels are closed. And therefore, if these voltage gated calcium channels are closed because my membrane is hyperpolarized, there is no calcium moving into the cell, there is no secretion of insulin. So when I have low glucose concentration, I therefore am not going to open my voltage gated calcium channels because I'm not going to release the insulin, because insulin is about lowering the glucose concentration further. So we need to ensure that our blood glucose concentration is maintained at the correct level. Now, insulin's not secreted because we haven't reached a threshold value. We've gone further away from it. We therefore have not opened our voltage gated calcium channels. So the resting state is where the cell membrane has this very delicate balance on either side of sodiums, potassium, chloride, calciums. So it's important when you get that question that you look at what they've told you in the stem of the question, because they might not link it to potassium, but they might tell you that it's something else which is involved in the secretion of a different hormone. So do make sure you're reading that, but it's really all about checking your knowledge of, do you know what it means when it's talking about a potential difference and whether something is depolarized or hyperpolarized? So those key words are the same regardless of what we're looking at. So I did a sample, I tested my blood sugar levels. So normally my blood sugar levels should be about 90 milligrams per 100 centimeters cubed. That's 4 millimoles per liter before a meal. So straight after eating a meal, I took my blood reading, this was a while back, when you used to have to know about biosensors. They've removed that from your exam this year. However, they may ask a question where they tell you about it. What it means is that you're not expected to memorize that if they've removed it, okay? But if I'm measuring 5.4 is my level, then what happens 2 hours after eating a meal? My glucose levels have risen up, 7.3. So because I ate the meal, because my body digested it, because I have the enzymes that are able to digest my polysaccharides down, because I'm able to absorb my monosaccharides into my bloodstream, my blood sugar levels have risen up. So 2 hours after eating a meal, they rose up to 7.3. Now, obviously, if my levels get too high, that's going to have an effect on osmosis. So it actually links quite nicely, Sarah, to what you said earlier with regards to the reabsorption of water. If I've got too much sugar in my bloodstream, that's affecting my water potential. That could result in more water being reabsorbed when actually I don't want the water to be reabsorbed. What I want to do is I want to get the sugar, I want to store it for a later date, or I want to utilize it and use it in respiration. So what happens in the beta cells is if there is a high glucose concentration, that glucose concentration can move across the membrane because it has a channel in there, differentiation of a cell. The glucose has diffused from a high concentration to a lower concentration. That glucose is then hydrolyzed down, glycolysis. Then we have the link reaction, then we have the Krebs cycle, then we have oxidative phosphorylation. We've now produced ATP. That ATP can now bind to my potassium ATP channel and they are now closed. If my potassium ATP channels are now closed, my high concentration of potassium inside is no longer leaving. So therefore, what we've got is we are now depolarizing the membrane because those potassiums are being moved in, but they're not diffusing out. So we're moving towards the threshold value. And if we reach the threshold value for those voltage gated calcium channels, they're going to open and the calcium will be able to flood in. Now, the calcium flooding in therefore causes these vesicles with the hormone, in this case insulin, to be able to move towards the membrane. And therefore, when they move towards the membrane, that is therefore going to result in exocytosis of the insulin out of the cell. So therefore, we have secreted the insulin out of the cell so that we can then have our blood sugar levels be brought back down. So remember, 2 hours after eating the meal, it went to 7.3. 4 hours after eating a meal, my blood sugar levels have dropped down again. And that's showing you, in this case, that my beta cells have released insulin, and the insulin has caused the blood to be removed. No, it hasn't. That's a lie. It's caused the glucose to be removed from the blood. And this is the thing that I keep saying this, and I said this with you last time, in the fact that sometimes you're thinking one thing, but you start writing it the opposite. And my brain is always going off in tangents, thinking about what else I need to bring in. And so therefore I have to take the time and read my answer. No, you don't need, there's not an expectation of talking about that, Lois, with regards to your answer. The key point is them looking potentially at, do you understand that the mitochondria are present? Do you understand that that is required for the production of ATP? If they want to go down the route and ask you about a glucokinase, which is an enzyme, they will give you a bit of information on that if there is an expectation that they want you to talk about that at all. And so therefore, just to go back here, if my blood sugar levels increase, then my beta cells secrete the insulin. If my blood sugar levels decrease, then it's glucagon. And I always think of that as glucose, agon. Glucose, get out of here. So therefore, when they bind to receptors, those receptors are at the target cell. That target cell is wherever whatever needs to happen is going to happen. So if it's insulin, we want to absorb the glucose. We want to have cellular respiration increase. We want conversion to glycogen. We want to form those glycosidic bonds. Or we may actually convert the glucose to fats, which is lipogenesis. And obviously the glucagon do the reverse, go the opposite direction. Right, let me answer this question. When taking repeats and calculating means and standard deviation, do you still include? Right, if you're calculating your standard deviation, then you are including your anomalous results. You do not remove anything. If you are just calculating a mean, you can remove anomalous results before calculating the mean, but if you're using standard deviation, you keep them in because that accounts for those means. What sort of cells absorb the excess glucose? All cells. So all cells, so when we are talking about, oh, sorry, which cells absorb the excess glucose? In that case, we're talking about the glucagon. So hold on, because I'm jumping from one. What sort of cells absorb? Yes, if we're absorbing the glucose, that's any cell whatsoever that has the need of glucose to be able to respire, that has the enzymes to do the conversion. So every single cell will be able to absorb glucose. But remember, we want to ensure that the glucose is stored. If we have too much of it, then the storage actually gets changed into the lipids that get stored in our adipose tissue. So it's important to note the difference between whether we're talking about the glycogen storage, so the glucose being converted to glycogen, and that's all cells as well as specifically your liver. It's one of the main roles of the liver, but it can be any cell. But if we can't store it as glycogen, we will then result in converting it into the lipids and store it as our lipids in our adipose tissue. Will you be marked wrong for including an anomaly in a mean? No, you should include the anomaly in the mean. The only way you wouldn't include the anomaly in calculating the mean is if there's nothing about calculating the standard deviation. So if it's an exam question and they have asked you to calculate the mean, there will be an expectation that you will include the mean because you're going to work out your standard deviation. If they don't want you to work out the standard deviation, then they will expect you to remove the anomaly. So you will need to be sure that you are reading the question to see, have they mentioned about the standard deviation, or do they later on ask you about an anomalous result? And if they want you to identify an anomalous result, it will be very obvious that it's an anomalous result. Sahib, back to your question. Do the calcium ions move down? Yes, they do. Sahib, they're going to move down their electrical chemical gradient, so you can make an assumption if they're moving into the cell that there was a higher concentration outside compared to inside. Absolutely. Does calcium ions bind to vesicles with insulin, then bind to the cell surface membrane? It's really interesting. So the calcium ions bind. If you think about cytoskeleton, what is your cytoskeleton made of? So your cytoskeleton can be microtubules or microfilaments. Microfilaments, sliding filament theory, actin. So what we have is the calcium binds to the troponin that is attached to the tropomyosin that moves the tropomyosin out the way. So what they will want you to note in this question is that the calcium is going to move in, and that is going to cause the vesicles to move towards the membrane. It actually is to do with it binding and causing the cytoskeleton to move them towards the membrane. So it's about linking those together. But they will only want you to go into that detail if they've given you some hints in the question. So don't get confused and start saying the calcium's binding to the troponin, moving the tropomyosin out of the way, because that's the skeletal muscle knowledge. So what you want to say is that calcium is binding to the cytoskeleton, causing the vesicles to be moved towards the membrane. Can we do a really quick look at secondary messenger? Yes, I do want to do that. That is my next thing I'm going to go on to with the secondary messenger. And statistical tests, you keep them in. Yes, absolutely. With statistical tests, you need your standard deviation, therefore you keep them in. Will you be going over plant hormones? If I don't have time just now, Julia, on plant hormones, I will try and go. I'll come on later, probably not until about 9 o'clock tonight, after I've done my group tuition session, to help. I know how close we are to the exam, so I'm trying to give you guys as much as I can with the timing that I have. Right, let me just cover the hormonal side of things. So I really do have a strong feeling that there's going to be something on hormonal communication, and it's going to be about, let me just find the thing I want to show you. About whether they do it in paper one. If they do it in paper one, it's going to be about the action of the hormones being a primary and secondary messenger. If they do it in paper two, it'll be about gene regulation. So it could come into either of the two papers, which is why I really feel like it's important that you do know the content here. So this isn't from your exam board, but it's a lovely question, which is why I want to use it. So in this question, in this exam board's question, they actually gave a really lovely diagram here, and it was talking about steroid hormones. And so therefore, when we're looking at a hormone, we're being told in the question, always take your time and dissect the questions. If I can give you any skills on application, a lot of the time the exam papers give you hints if you find them. So you're told it's a steroid hormone that's involved in cell signaling. You are told that this figure is showing you the sequence of events. They want you to do one of two things. They want you to explain why hormone S does not need to pass through a transport protein. So we can already see that hormone S is moving through. Well, they gave it in the picture, the cell surface membrane. So therefore, what they're actually asking you is, do you know that a steroid hormone is non-polar? Do you know that it can cross the phospholipid bilayer because it's not repelled by that hydrophobic region. Yes, absolutely. That's the key way in putting it. You should really understand that there are many different ways. There was 20,000 or 22,000 of you who sat the exam last year, and therefore not everyone's going to say it's non-polar. Some of you will use the correct term that it is lipid soluble. Because it's a lipid soluble hormone, again, it's the same thing. It can cross the phospholipid bilayer because it is not repelled by the hydrophobic region. There you go. Absolutely. Same question that's just come through. But yes, absolutely. It's to do with the fact of they're checking your understanding, but they know that some people will say lipid soluble. They know that some people will say non-polar. They won't penalize you. So what they'll show it is lipid soluble slash non-polar slash hydrophobic. Any of those three wordings will allow you to get that marking point. No, 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 no. It's not two. That would only get you one because the lipid soluble part of it is just you naming about the molecule. The fact that it can cross the phospholipid bilayer because it's not repelled by the hydrophobic region is the second marking point. So it's important that it can pass the hydrophobic core because it is hydrophobic itself. So you need those 2 points to get the marking point. So it's important that and that's what a lot of students do in the actual exam. They're like, oh, I said that. It's non-polar because it can cross the phospholipid bilayer. Well, all you've done is rewritten the words that are in the actual question. It's non-polar. It can cross the phospholipid bilayer because it's not repelled by the hydrophobic region. So it's really important when you are doing these types of questions and when you are answering them in the exam that you do say every little bit of detail that you can. Don't assume that you have told me, the examiner, what you know by saying it's non-polar, so it can cross the phospholipid bilayer. I don't know what you mean by that. I don't know if you actually understand what that means. All you have done is you've reworded that. Well, in a sense, you've not reworded that. You've reworded that it can pass through. So that's why I would say always make sure you try to be specific in your answers. And that's where a lot of you struggle with when you say, oh, the mark schemes are so specific. They are specific. They have to be because they've got to be prescriptive to us as examiners so that we can then actually know whether we'll accept or not accept the mark scheme. Now, you won't see the extra notes that examiners are given, just so you're aware. As examiners, we are given extra notes that even teachers don't get to see because they're not marking the exams. So it's important that you know that there are some variations, but it's not something that they would publish out there because one year, if they publish it and someone writes it again, they might not accept it because it's a different set of examiners who are marking it. So the things that are in, excuse me, the box in a mark scheme, they are absolutely specific wording that they want you to say. So therefore, they want you to know that that is what's acceptable. If you were to regurgitate that specific to the question in front of you, you will get the marks. But if you just memorize, you will lose marking points because a lot of the time it's about relaying it to the question. So let's look at this question as well. The target cell responds to other cell signaling molecules in addition to hormone S. The cell has, and this is interesting, receptors in the cell surface membrane, in the cytoplasm, and in the nucleus. So we have these receptors in different locations. So explain why hormone S binds only with receptor R in the cytoplasm and not with the other receptors shown in figure 3.2. So this is an important thing of using your information and also using the knowledge of what you have got to be able to figure out what they're telling me. So we have got this receptor, which is R. So because we've got this receptor, which is R, what we know then is that receptor is obviously going to have a very specific shape. And that shape is determined by the fact that it folded in a very specific way. So if we're looking at a question like this, we have to understand that it's not binding to the receptors on the surface because it's not a peptide hormone. We know it can get through, and because we know it can get through, we know that it will only bind with receptor R because receptor R has got the shape which is complementary. Oh, wrong pen. Complementary shape for receptor R because they will form a hormone receptor complex. So it's important when we're looking at this that we really do check our knowledge. What are they asking us? Well, what's a receptor? A receptor with a very specific shape for the hormone that it is going to bind with. Yes, they could enter the nucleus, Lois. It all depends on whether or not the receptor is in the nucleus or whether or not the receptor is in the cytoplasm. Do I have any tips for revising the PAGs? The PAGs basically are kind of irrelevant at the moment. And I say that because the PAG that you sat for one, PAG one, the microscopes, is you using a microscope and you learning how to calibrate the graticule and you observing and drawing the cells that you saw, which were mitotic cells. That PAG was for you to practice using those practical skills so that they can say whether or not you get the endorsement. What's important when it comes to the PAGs is going through the specification and looking in modules 2, 3, 4, 5, and 6. Where are their practical skills that they will be assessing? So, for example, microscopes is in cells, the cell structure topic area, but actually they could talk about the blood cells, which are in transport in animals, and using a microscope to observe them. They could give you histology, which is to do with the different muscles, and expect you to use that. They could look at plants. It really doesn't matter. It's not one size fits all. The PAGs were there to give you the skills to then be able to apply that to the exam. So if you're doing a practical or you're given a practical, can you basically tell them what's the independent variable? What's the dependent variable? What are the control variables? What equipment should they have used? What are the uncertainties? How would you improve this? That's the skills that you are going to get from the PAGs that you then bring into the exam. So that's why it's module one. So module one is about those. Are there any required practicals? No. It's not like AQA. AQA has required practicals that they have to do. OCR have got, you must do 12, so PAGs 1 to 12, but the teachers can pick and choose what they decide to do, but the content you'll be assessed on is in your specification. Could you go over calibrating the microscope? Lois, I did do that in my live yesterday. So if you check the video on yesterday, obviously you can skim through. I did do a bit on calibration yesterday, if not yesterday, the time before, but it's definitely on my live already. And I have a video on module 2.1.1, which has how to calibrate. It's an interesting one. The 6 marker evaluation questions could be on anything at all. But let me see if I can come up with, let me see. Let me just randomly pick one and go through how we could do an evaluation. Go on this. This is I usually always mention stat says sample size and then I just find it. Yes, absolutely, Shane. Absolutely. That is exactly what you need to do. You need to be looking at how they have controlled it. So the other thing I would add to your list is control variables. Have they controlled every bit of the variables that they could have controlled? If they haven't, what are you going to can that spell? What are you going to suggest that they do? All right, here we go. When was this? This is a random specimen paper. Now, I always say to students to, whenever they are doing any type of practical, any type of revision, also do the AS papers because the AS papers are really good for helping you on that one. Need to just do an agree, disagree, bullet points. Yes, absolutely. As long as it's logical and as long as you have followed what needs to happen, then yes. Um, can you please cover DNA replication, conservative and non-conservative replication and the experiments for them? Harrison, if you go check out my nucleic acid nucleotide playlist, you'll see stuff on that already. Depolarization in neuronal communication. Do you say sodium ion diffuses in down electrical chemical gradient? Yes, it can be down the electrical chemical gradient, or you can say down the concentration gradient. They're actually not very strict on that one unless they've specifically mentioned electrical chemical gradient before, then they will want you to say that, but they will allow both normally. It's normally they want you to say electrical chemical gradient, but if you said concentration gradient, they will then give you potentially a mark. Okay, let me just go through this one a second. So here is a random one. Um, so Lois, just to go back to your thing that you've said, you didn't realize you need to know experiments for them. You need to go and look through the specification because there is very specific ones like the TLC for amino acids and the TLC for chromatography. They're for chromatography for photosynthetic pigments. You need to know they could ask about either of those. A dissection could be on anything. It could be the heart, could be the kidneys, could be a plant. Stem, it can be anything they want it to be if they do a dissection. There's not one dissection you need to do. It's whichever one, which actually let me just put up. So what I would always say to you is make sure you are using the specification as you go before I answer this question. Hold on, bring it down here. So this is any part that they could ask you, which I use as my guide. I go back up here. So if you look at this one, then you can see appropriate apparatus. So they can ask a question on any of that. So do you know how to record a range of quantitative measurements? So do you know what mass, how to measure mass, how to measure time, volume, temperature, length, and pH? Do you know the appropriate instrumentation to record the quantitative measurements? So do you know colorimeter? Do you know potometer? Do you know how to use a lux meter? Do you know how to use? So these are all things that you should be aware of. A pH probe, an oxygen probe, a carbon dioxide probe, universal indicator. It depends which way they want to go. So these are skills that they have assumed that you've gained in GCSE and into A level, but there's no you must do this or you must do that. It's that we expect you to be able to get these recordings by doing both. So last year they had transpiration and a pedometer and also colorimeter. So it really just depends where they want to assess you. Light microscope, they can give that in any place. Production of scientific drawings, anywhere. So these are skills that they expect you to have learned throughout the 2 years whilst you were doing the course. Oh, sorry, you meant the conservative versus non-conservative. Well, that's an interesting one, actually, on that one, because let me just find it. It used to be, it's in the book, and this is where, again, it's really important that when you are doing your revision, you are using the specification to help you, not just using your textbook, because the textbook is good, but the textbook will give you much more information than what is necessary. So if you look here, the only one that they are actually expecting you to know is semi-conservative replication, and to understand how that actually works. So then you look at this and bring those together. So do make sure when you are doing these, oh, that's interesting. Why did that do that? That you are looking at what it is actually saying in the specification, because that's the important part. That's the one you have to know. If they're going to ask you about the conservative replication, it will be a distinguishing and they will have given you information about it in the stem of the question. So it is important that you are doing your revision, but you're using the specification to help you. That is what they have agreed, that this is what they will assess you on, but they could apply it to a concept which is slightly out of your norm, and therefore you should be able to use that. If semi-conservative replication is where I have one strand, which is the original, and one new strand within my molecule, then conservative would be where I have the original and then I have the new strand as its own thing. So it's important that you are, one, making sure you're following your specification. OCR and AQA are different inset things that you have to know. AQA don't need to know about photophosphorylation, for example, those terms, but they need to know about the process. So do make sure you are using your specification and not just revising stuff that other people think is in it, because they have given you a video that's OCR, but actually it's also a bit of AQA in there, would be my little bit of advice. Um, and then back to this question then. I'm going to leave this up for one last second. I'll be back in 1 minute. Okay, back. Right, this question then. When we are looking at this question, what have they told us? So I've got an investigation. I'm looking at the effect of pH on the activity of amylase. So straight away, I would just give myself a little trigger that says that here's my pH. I know that I'm going to have an optimum, and anything away from the optimum is going to cause denaturation. So therefore, we're going to get a decrease either side. So that's the first thing I do is I give myself a bit of information as I'm reading the question. So this was the method that was used. Tubes containing starch. Yep. So we think the starch is going to be broken down by the amylase, which is the enzyme. We're set up in a range of pH buffers. They use the same concentration of starch and amylase. Okay, but did they use the same volume? So they've controlled the concentration, but they haven't used the same volume, or they have. I don't know. They haven't told me. Were used each time. A small sample of the solution was removed. What does it mean by a small sample? How much? Was removed and tested for the presence of starch. Okay. They're testing for the presence of starch, so therefore they've used iodine solution. How much did they use of the iodine solution? And they did it at 20-second intervals. So I'm assuming that they have got a white towel, a spotting towel, that they are then going to put their samples on. So a spotting towel. Right, okay, the students present the results in tables 2.1, right mean, amylase activity percentage of the maximum. So you can see we've got a low number here, we've got a low number here, and it's rising up to 100%. So therefore, I would assume that 7 is my optimum. Just looking at this, I'm not seeing any of the questions. I'm just looking at this right now, and I'm trying to say to myself, right, well, what could it possibly be? And then I'm saying, well, actually, 96% and 100% is actually really close, and they haven't told me anything about my standard deviation. So I don't know about the standard deviation, so I don't know if 6 or 7 is actually the true optimum. So I would probably want to do small intervals between the two. Oh, look, and there it goes. So the student concluded that the optimum pH for the amylase was pH 7. A teacher made the following statement. The results in table 2.1 provide only weak support for the conclusion. That's true because they follow the trend, and that the optimum pH for amylase is pH 7. Evaluate the statement and suggest improvements to the student's procedures that would support the conclusion more strongly. So that's where I would say what you need to include and think about is, well, do we know what the volume was? Do we know how much of the different solutions that they were testing? Can we be sure? We've got no standard deviation. So that's the sort of thing when it asks you to evaluate something, it's basically pull it apart and find faults in it, because that's going to help you to maximize your grades. There's no one answer that's correct for this. It's about what you can find when you look at that. So I hope that's been helpful. And yeah, I hope that it's important that you understand that you want to take the time to read the information. I will try and jump on here a bit later again, unless anyone's got specific questions. Could you talk about a lack of control here? What would your control be? So, Tony, what would your control actually be if you were to do this? Because if you're doing this, you'd have starch and amylase, or you'd have starch and distilled water, sorry, with no amylase, but you wouldn't get any results. So I would say that that wouldn't improve this one. Doing it in smaller increments definitely would, but not actually the use of a control, unless you were going to compare the difference between the two. And you were talking about using a control to compare, but I don't see how that would help in this case, apart from just proving that you need the amylase for it to break down. would be what I would suggest. What was the questions? Do you think they could ask about the difference between parent? Yes, I do believe that. I do feel like a T-test is coming. Yeah, really, the only improvements is about repeating it in more intervals. Absolutely. For you to be sure, that's the only way to really improve this practical. Yeah. But Shane, back to your question. Yes, they could. So the important thing is when you read the information in the question, are they talking about the same individual and therefore the resources being linked, or are they talking about it being unpaired? So in this case, this would be if we were to look at the pH 6 and 7 and to see if there was a significant difference, we would do an unpaired t-test because 6 and 7 is not linked, so therefore it's not paired. And we would do the information, but to be able to do the t-test, we'd need the standard deviation, and we don't have that information in what they've told us. This was actually an AS paper question. Um, yes. So, Lois, that's a good thing with regards to temperature. What about temperature? They haven't told us anything about temperature. So, the question really is about how evaluate the statement and suggest an improvement to the student's procedure that would support the conclusion more strongly. So to be able to support the conclusion that pH 7 is the optimum, you want to do more intervals. If they just ask you to evaluate the investigation, that's when you bring in about the control variables like temperature. So it's really important you're reading the question that's been put in front of you. Whenever I go through these questions, whenever I am highlighting and analyzing, I'm not looking at what the questions ask me yet. I'm putting all the different points that I can find in case they ask me that question. So for this evaluate one, which says that would support the conclusion more strongly, that's about can you be sure that 7 is the pH, the optimum pH for the amylase? And therefore, you want to do more between the intervals. Would it be worth going all the way down to pH 8? Which is an interesting question. So, should I actually say to do intervals between here and here? Is that a good? It drops after 8, but what if it was 7.1? And it didn't drop until 7.5 and it dropped dramatically at 7.5. It's an interesting one. They would have accepted all the way from 6 to 8, but I wouldn't have gone as far as that because quite clearly you can see it's somewhere between 6 and 7, but it may have been from 5.5 to 7.5 would be a good set of values to do because 6 is 96. Well, actually, what if it was 5.9? So again, it's about making sure the answer that you give is relevant to the information that you've been given in the exam and not that you've memorized something. That's the bit as well that most students kind of drop marks at, is that they will go to an unrealistic point. Because if I'm going to go all the way to 8, surely I should go to 5 because 5 is not as big a drop. So therefore, it's about what's realistic. Let me get rid of all this when I'm doing my answers. How did they measure the amylase activity? So my assumption would be to measure the amylase activity, they'd be looking at the iodine. So they would have started with a tube containing starch and iodine solution. So it would be blue, black in color. And then what they would then do is take sample, well, actually either or. So either taking the samples and putting it in the spotting tile and then putting iodine there, or they could have it that it's got the iodine already in it and they are taking samples to do a colorimetry on it. It really just depends what they decide. But again, it's you using your knowledge, and as long as that knowledge is correct, they won't penalize you for coming up with a different way of doing that measurement as long as you know how it would actually work. Would a colorimeter be a suitable way of making it quantitative? Yeah, so a colorimeter would be a better way of making it quantitative because then what you'd be doing is you would be taking a sample, putting it through the colorimeter, looking at the absorbance. So yes, that would be. If you were to do colorimetry for this experiment, what colored filter would you use? So in this case, oh, interesting. So we're going to do iodine, which is a blue-black color. So I would say blue-black if starch is present. It's orange-brown when it's a negative result. So I'm looking at, if you think about it, when you're using the colorimeter for the Benedicts, you use the red filter, and therefore, because you're using the red filter, it allows the red to go through. So I'm just thinking through. Yeah, saying complementary colored filter for the colorimeter would be one, but it's a good question. I don't actually know what one I would use, to be honest. Hold on, let me look it up. Um, red or orange filter? No, because we've got the blue. Hold on. Looking at orange. Use a blue filter. Blue filter. Final answer, blue filter. And that's a hard one because actually, if we're looking at Benedict's, we're using the red filter. But if we're looking at this, we're looking at the blue filter because that's why it's about the filter of the positive that's going to give you the positive color. It's a hard one, but if they're wanting to give you information on that, if they're asking you, they will tell you unless it's the obvious one, which is Benedict's and it's red. So what I would say to you is go with a answer which covers them, a complementary colored filter for the test that you're using. That would therefore give you the marking point because you've not actually stated a wrong color. You've said you're going to use one which is complementary, and therefore you've shown that you have to think about the color. So the blue is because if you think about the positive result for starch, when iodine, you use the iodine test for starch, it goes blue-black color. It doesn't state it. You should know that. You should know if you're testing for the presence of starch, you would do an iodine test. That's a you should know. What's the difference between transmission and absorbance in colorimetry? It's to do with how much. So let me just. Oh no, didn't mean to do that. 1 second. Let me go this. This is kind of where your GCSE knowledge needs to come in. Um, the if you think about if I've got my light source and then I've got my curvette, what I'm going to do is I would have my filter in between. My light's going to come through here, and that would be white light. Then whatever color filter you're going to use is going to allow that wavelength to go through. If we're talking about transmission, we're talking about how much of it has actually been transmitted through. If we're talking about absorbance, how much of it was actually absorbed by the molecules which were inside the curvette. Yeah, no, I changed my mind. That's why I changed it to blue. Yes, yes. So if there is more absorbance, then you are talking about, so if you were looking at, let me get you an example. So if we look at this question, I'm not really looking at the question, I'm just looking at the graph here right now. What you can see is you have got a concentration of enzyme and you've got the absorbance. So what you can see is as I increase my concentration of enzyme, the absorbance has also increased. So, so if I've got, for example, a concentration of enzyme which is catalyzing the reaction, it's going to break down the starch into the sugars. Then I'm going to have an increase in the absorbance because there's more sugars present. So it depends what you're looking at and what your test is. So if it was the iodine test we were looking at, and the iodine test, if there is more absorbance, there would therefore be more absorbance, more starch present. Less absorbance, less starch. If we were looking at Benedict's, we would therefore say that the starch has been hydrolyzed down into the glucose, and therefore we have now got it where there's more. So if there's Benedict's more absorbance, then we would therefore say that we've got more glucose or reducing sugar to be more precise. Sugar. Does that make sense? So it really depends on the question and what they've told you because it can be adapted any which way. Yeah, Lois, wrong way round. Yes, it is. It's a weird one with the proteins one, but yeah, you're going to use a green filter on that one. The only one you're meant to know about actually for your exam is the filter which is closest to the wavelength of that that's going to be absorbed. So when you're looking at anything to do with colorimetry, it's all about wanting a complementary color of the solution. So the color of the solution absorbs the most strongly, and that's what's important. So if we are using the iodine, we're going to use a blue filter. The starch obviously being broken down by the amylase means that the blue-black color will decrease over time, and that's what you would use for that one. Pancreatic duct and blood vessels in an electron. Oh, okay. So a pancreatic duct, good question. Time are we on? 2 o'clock, right? Close to two. On that one, if you're looking at the duct versus the blood vessel, you've got to remember that a blood vessel, whichever blood vessel we're looking at, is going to have your layers to it. So you should have 3 layers to your blood vessel, whether it's a vein or whether it's an artery. Whereas when you're looking at a duct, like the pancreatic duct, like the bile duct, that's just going to have lots and lots of cells and not have these layers. These are my really dodgy cells. So you wouldn't have the layers like you would have the layers in your blood vessels. So you'd be looking for just lots of cells Lots and lots and lots of cells would be a way in which you would distinguish between the two. So the starch would be absorbing. The right is meant to be a duct. So this could be the pancreatic duct. It could be the bile duct. Doesn't really matter what it is. What I'm basically saying is that you have a duct that is lots of cells which are going to release into, because they're the exocrine system, they're going to release their digestive enzymes, let's say if it's the pancreatic duct, or if it's the bile duct, it's the hepatocytes which are releasing the bile into the duct, the bile duct. Guys, I hope you've enjoyed this. Good luck. Keep going. Keep practicing and looking at as many past paper questions, and know that you can't memorize it all. They will throw curveballs at you, and even I will have to sit and think when I see these papers, as will your teachers, to think about what is it that they're looking for? What did they want to get out of you? That's the important part, is that we don't know. Just like you don't know, we can look at the question paper and go like, oh, I think they might be asking that, but we won't know for sure until we see the mark scheme as well. I will be live tomorrow evening after I've done my session with my clients. I will try, I'll hopefully be back on here at 9 o'clock.